This is Chapter 2, Chemistry, Part 2, Organic Chemistry. Uh, these are the second part of the slides on organic chemistry. We're going to go through the slides. Organic chemistry is related to molecules, okay, that contain carbon and hydrogen. Remember I told you compounds and molecules were used interchangeably. So here's an example of talking about a molecule that contains carbon and hydrogen. So organic chemistry deals with molecules that contain carbon and hydrogen. There are 4 types of organic molecules. They're carbohydrates, lipids, proteins, and nucleic acids, and we'll go through each in detail. So organic chemistry deals with a number of things, but specifically we are dealing with these macromolecules. These are the molecules of life: carbohydrates, lipids, proteins, and nucleic acids, both in animal and plant life. Carbohydrates contain carbon, hydrogen, oxygen, usually in a 1:2:1 ratio. Carbohydrates contain carbon, hydrogen, and oxygen in a 1:2:1 ratio. They are usually in a ring, okay? This would be a ring here, and everywhere there's a junction here where these points come together in this ring, there's a carbon. So this is 1, 2, 3, 4, 5, 6. This is a 6 carbon ring. You will not have to draw structures. You will not have to memorize structures. You do need to know that a carbohydrate is in a ring. They are covalently bonded. That means they share electrons, which is a very strong bond. Primary function for carbohydrates is quick energy supply for the body. Classified by the size, if you have a single carbohydrate, it is called a monosaccharide. A single carbohydrate is called a monosaccharide. It is made of carbon, hydrogen, and oxygen in a 1:2:1 ratio. This is glucose. We know glucose. That is the source of plant photosynthesis. Plant photosynthesis takes carbon dioxide and takes water in the presence of sunlight and makes glucose and oxygen, and we take then that glucose in the presence of oxygen and break it down into carbon dioxide, water, and ATP. This is fructose. This is another monosaccharide. This is galactose. This is another monosaccharide. These are 6 carbon rings. Here are an example of 2 monosaccharides that are 5 carbon rings. The first is deoxyribose. You've heard of that from DNA. That's deoxyribonucleic acid. And ribose from RNA, ribonucleic acid. The only difference between deoxyribose and ribose, if you look here at this, this side branch here, this has an OH, and this only has an H. So this has deoxyribose. So these are the most common sugars that you will deal with. There again, you don't need to know the structure. You will not have to draw the structure. You will not have to recognize that as glucose by sight, okay? Glucose, fructose, ribose. Monosaccharides have from 3 to 7 carbons. However, the ones that we deal with have 5 and 6 carbons. 6 carbons are called a hexose, and 5 carbons are called a pentose, okay? Glucose and fructose are 6 carbons, but in different arrangement. Ribose and deoxyribose are 5 carbons. So the ones we deal with most are the 6 carbons and 5 carbons. Two monosaccharides will join by dehydration synthesis. We've already talked about that, dehydration synthesis. We take an OH off of one side chain and an H off of another side chain. And when we do that, that joins these 2 carbohydrates together. Okay, so right here we've joined 2 carbohydrates together. We joined glucose and fructose, and that actually gives us sucrose, which is our table sugar. Glucose itself is not very sweet, but if we join it up with fructose, then fructose is fruit sugar, that makes it sweet, okay? And so that's table sugar right there, glucose and fructose. This is 2 glucose molecules joined together, and this is a galactose and a glucose molecule joined together. So we have sucrose, we have lactose here, and we have maltose. Those are those three three molecules there. You don't have to know you don't have to know the structures of them. Created by dehydration. Remember, dehydration removes water. Okay, it's called dehydration synthesis. We take an OH off one group and the H off the other group, and then they join together, and that creates a bond. They're ideal storage products. In plants, we store joined monosaccharides in the form of starch. In animals, we store joined monosaccharides, which are called polysaccharides. So multiple monosaccharides joined together are called polysaccharides. If you join two monosaccharides, it'd be a bisaccharide, trisaccharide, and then polysaccharide. There is no hard definition for polysaccharide, but starch is a polysaccharide. Long chains of monosaccharides, same thing with glycogen. Okay, this is a molecule of glycogen. This is a schematic of just part of it. This is a longer term storage product. Glucose is a short term storage product. Glycogen is a long term storage product and is in the muscles and the liver. Next is lipids. So we went over carbohydrates. The next category is lipids. We're going to look at triglycerides, phospholipids, steroids, and eicosanoids. Okay, triglycerides. It contains carbon, hydrogen, and oxygen, just like the monosaccharides, just like carbohydrates do, but they're not in a 1 to 2 to 1 ratio. Okay, they're not in a 1 to 2 to 1 ratio. Less oxygen is present. Okay, triglycerides most fats are not soluble in water because they don't have a charge on them. Okay, long-term energy storage and cell membrane component. We'll learn when we get to chapter 3 of the cell that we have phospholipids, a bilayer of phospholipids in the cell membrane. A triglyceride is composed of a glycerol backbone and 3 long chain fatty acids. So a glyceride is composed of a glycerol backbone. This is glycerol, and these are long chain fatty acids. And guess how we join them? Dehydration synthesis. We could take a hydrogen from one side, an OH from another side. We take this out, okay? And now we have a carbon-oxygen-carbon bond, okay? So we have removed water here. So we have done a dehydration synthesis, and now we have this glycerol backbone joined to these three long chain fatty acids, okay? And the water has come out, and that's called dehydration synthesis. We saw that in the monosaccharides. Now we see it in triglyceride, okay? Triglyceride has 2 possibilities. It can have a saturated fatty acid, okay? So this is a acid, COOH gives us the acid group, and this is saturated, which means that every one of these carbons after the acidic group has full complement of hydrogens, okay? One hydrogen, one hydrogen, one hydrogen, all the way out. So this is a saturated fatty acid. No double bond carbons. These are animal fats. These are solid at room temperature. These are currently not the good fats for you, okay? This is an unsaturated fat. All an unsaturated fat means is you have a carbon-carbon double bond. Let's go back to this other slide. Why these are so bad for you is, number one, they're linear. Notice they're in a straight line. You can pack multiple molecules on top of each other. So it becomes highly dense, becomes solid at room temperature, a lot of calories there, okay? With unsaturated fats, you have a carbon-carbon double bond. You have a carbon-carbon double bond. This carbon-carbon double bond, number one, results in a kink in the molecule. So rather than having that nice linear molecule that you put one on top of the other, like a page of a novel or a page in a textbook, this has a kink in it, okay? So you try to lay the next one on there and it doesn't fit perfectly. It doesn't fit perfectly. And so you have fewer of these per space, okay? And so this is a carbon-carbon double bond. And as you can see, before we had 2 hydrogens attached to every one of these carbons. Now we only have, where the carbon-carbon double bond is 1 hydrogen attached. That makes it unsaturated. These are your vegetable oils. They are liquid at room temperature. Currently, these are your good oils, okay? What we have done now is we have the ability to saturate an unsaturated fat. Okay, we have the ability to saturate an unsaturated fat. So what we've done here, we're looking at trans fatty acids. We have a carbon-carbon double bond. The carbon-carbon double bond would indicate that it is unsaturated, okay? And we only have one hydrogen there, one hydrogen there, one hydrogen here. But look here, we have, remember, carbon has typically 4 bonding places. We have 1, 2, 3, 4, 5. So this carbon technically should only have one hydrogen. But it has 2 because through chemical manipulation, we have the ability to add a hydrogen. So this is a trans fat, okay? So this is a unsaturated fat that has been resaturated. In other words, through chemical manipulation, we have added a hydrogen there, okay? Next, we'll talk about phospholipids. Phospholipids are very similar to the triglycerides. Let's look here. This is a glycerol backbone. We have 2 long-chain fatty acids. One here is a saturated fatty acid. One is an unsaturated, okay? So we have a glycerol backbone like before. We have 2 long-chain fatty acids, but here in the place of the 3rd long-chain fatty acids, we have a phosphate group. So it's called phospholipids. So we have a phosphate group, okay, phosphate group. This phosphate group is polar, okay? It has a charge to it. So it would attract a liquid. It would attract water. Water would be attracted to it because water is polar. This fatty acid, as you can imagine, is nonpolar, okay? It's nonpolar. So this tail here, you have a polar head. So we're looking at the molecule in a schematic here. There's its polar head, there's the phosphate group, and these are the long chain fatty acids, okay? The phosphate group is polar and the long chain fatty acids are not polar. So if this was in a cell membrane, this would point outwards. This would point outwards in the plasma membrane so that fluid, interstitial fluid, which is primarily consists of water and brings all the nutrients, brings the gases and everything to the cell, would be able to come right up to the cell membrane, okay, because it's attracted to this charge and deposit the things it needs to into the cell. Same way on the other side, we have a phospholipid layer. So we have 2 layers of this. So that we'd have 1 layer of this, and imagine a copy of this going outward like this. So on the inside of the cell, inside of the plasma membrane, we'd have a phosphate head here and 2 long-chain fatty acids coming in this direction. Same type of thing. The inside of the cell has a large amount of fluid in it, and the majority of that fluid is water. So we'd want to get rid of waste products, or if we had something we need to transport out, we'd want to be able to be drawn to the cell. So we have a biphospholipid layer, and we'll see that in chapter 3. So we have triglycerides, which have a glycerol backbone and 3 long-chain fatty acids. We have phospholipids, which have a glycerol backbone, 2 long-chain fatty acids, and a phosphate group. Covalently bonded to the glycerol backbone. Guess how they're bonded? Dehydration synthesis makes this phosphate group amphiphilic, okay, which means it's a polar and a nonpolar end, and it's needed to build cell membranes. Our 3rd group of lipids are steroids, and they have 4 carbon rings with side chains. So what you need to know about a steroid: 4 carbon ring, 4 hydrocarbon rings with a side chain, okay, has no long chain fatty acids on it, okay. Cholesterol is one of the steroids used to make vitamin D, steroid hormones, bile salts, corticosteroids. It's the building block. We make 95% of all the cholesterol we need. The additional 5% we need to take in, obviously, with our diet. In the United States, we take in well more than the 5% that we need to take in. The body makes 95%, so that we end up depositing cholesterol in a number of places, including inside our blood vessels. Eicosanoids are cell messenger, cell signalers, okay? influence cell function in a variety of ways, regulate blood pressure, smooth muscle contraction, mediate hormones, participate in blood clotting, and inflammatory reaction. That's all we're going to say about them, okay? Next group are proteins, okay? Contain carbon, hydrogen, oxygen, and nitrogen. So the basic protein, basic amino acid, which is the building block of proteins, just like monosaccharide was the building block of carbohydrates, amino acid is the building block of a protein. It contains carbon, hydrogen, oxygen, and nitrogen, and then some of the side groups of the amino acid. We have 20 different amino acids, and the difference in the amino acid is determined by the side groups. Some of the side groups have sulfur or phosphorus in them. The structural unit of the amino acid. What's the structural unit of the amino acid? Okay. Every amino acid, if we look down here, has a central carbon. Every amino acid has a central carbon. It then has a amino group. This is an amino group, NH2, amino acid, okay? And we have carbon, hydrogen, oxygen, and nitrogen. That's why all amino acids contain those groups. So we have a central carbon, and we have 4 attachments to it. One of it is an amino group, NH2. One attachment is a simple hydrogen. One attachment is a carboxy group, an acid group, COOH. Okay, this is our acid group. This is our amino group, amino acid. And then the other group, the thing that distinguishes the 20 amino acids is the R group. Okay, the thing that distinguishes the 20 amino acids is the R group. And the R group is different. You need to know the basic structure, so you need to know this structure right here. The centralized carbon, the amino group, NH2, the acid group, COOH, a single hydrogen, and then an R group. The only one I remember is glycine, because in the R group is a hydrogen. The rest of them I don't remember. So I don't expect you to remember anything except for this basic structure. Know there's 20 essential amino acids, and the difference between the amino acids is what the R group is. Proteins are made up of amino acids. There's 20 types. All have amine groups and acid groups. R groups make them different. R groups make them different. So this is glycine, the one I remember because it has a hydrogen. This is aspartic acid. This is lysine. This is cysteine. They all have different R groups. 20 different amino acids. They have different R groups. This is why we can eat animal protein. The animals we eat have the same amino acids in their protein. We don't use their proteins. What we do is digest protein down to these are 20 essential amino acids, and then we use those amino acids to build our proteins. Amino acids are joined, guess what? Dehydration synthesis. Can you see now the importance of knowing what dehydration synthesis is? There'll be a question, at least one, maybe two. It could be a short answer question on dehydration synthesis. Explain in detail what is dehydration synthesis and where it occurs. Well, already so far, we've seen that it occurs in carbohydrates, we've seen it occurs in lipids, and now we're seeing that it occurs in proteins, okay? So we're gonna remove a hydroxyl group off the acid and a hydrogen off the amino group. When we remove a HOH, that's H2O, water is gonna come out. That is going to join this, okay, with a carbon-oxygen-carbon bond, covalent bond. Guess what we do if we want to, once we digest proteins, we're going to add water back and energy and enzymes, and we're going to break it apart. Okay, so that would be a hydration reaction. But to join these things, which is what we're considering now, we use dehydration synthesis. Okay. Changing a single protein in a long amino acid linkage, which is probably will result in changing the characteristic of the protein. We'll get into that in a little bit. Why? There are 4 levels of structure. I can envision this being a short answer question worth 5 or 10 points. So I can envision define and demonstrate with words what are the 4 levels of structure of a protein. First level of a structure is a list of the amino acids starting with amino acid number 1, going through amino acid number 1,000, maybe 5,000, whatever it is. Starting with amino acid number 1 and going through the list till you get to the last amino acid. It's like your class roster, starting at A and going to Z. So primary structure is just the names, okay, of the amino acids starting at one end and then into the other. It's very easy. Primary structure is the name of the amino acids starting at one end and going to the completion of the molecule and naming them in order. Notice we have R groups here. Okay? And also we have some hydrogens and nitrogens. Nitrogen is also one of those molecules, okay, that can undergo hydrogen bonding as well as oxygen is, okay? So notice that we have some nitrogens here, we have some hydrogens here, we have some oxygens here. Our primary structure in review is a list of the amino acids from amino acid number 1 to amino acid number 50, number 100, number 1,000, whatever it is. That's primary structure, linear structure. Secondary structure refers to geometric arrangement. It either can be an alpha helix, or a beta pleated sheet. An alpha helix or a beta pleated sheet. These are the two secondary structures, and they are related to the geometric arrangement of hydrogen bonds. So with these hydrogens here, with these oxygens here, with these nitrogens here, we have some potential depending on what the R groups are. The localized, if maybe one of the R groups is a hydrogen, okay? Maybe one of the R groups has a electronegative atom in it. But we have the potential for hydrogen bonds, okay? Locally in the area, okay? So we're talking about, and I'm making this up, but maybe there is a hydrogen bond between amino acid 1 and between 20, and maybe between 25 and 50, okay? We're not talking about a bond between amino acid number 1 and amino acid number 1,000, okay? We're talking about local, okay, hydrogen bonds, which result in either an alpha helix or beta pleated sheet. And the determination of that is the primary sequence of amino acids will determine what the secondary structure is based on which amino acid is there and the characteristic of that amino acid. So secondary structure, they go to a geometric arrangement by way of hydrogen bonds. They either are in an alpha helix or in a beta pleated sheet. A beta pleated sheet is just like a piece of paper that if you ruffled up and it had 4 or 5 pleats in it, that's a beta pleated sheet. Tertiary structure is an interaction between the R groups at a distance. So it might be between R group number 1 and R group number 500 and R group number 750 and R group number 1,000. So it starts to look like this. It starts to fold on itself to get this tertiary three-dimensional structure, okay? Three-dimensional structure. And this folding is very specific based on the linear arrangement of the amino acids. That's why that statement about several slides ago says that if you change a single amino acid, we may not end up with a structure here. If we don't end up with a structure here, we don't have a functional protein. That's very important to understand. If we do not end up with a structure, we don't have a functional protein. So this is the tertiary structure. We now have a three-dimensional protein. Looks like, to me, a can of worms or something like that. But these are interactions between R groups at a distance. And finally, quaternary structure, we take 2 or more of those tertiary structures and we put them in one big molecule, okay? The quintessential example, teaching example of that is hemoglobin. It has 2 alpha chains and 2 beta chains. So it has a total of 4 chains, okay, of these tertiary protein structures. 2 of them are alpha and 2 of them are beta, and they come together to form a quaternary structure. So those are the 4 stages, the 4 structural things of protein: primary, secondary, tertiary, and quaternary. And you're going to need to know those because there's going to be a short answer question coming your way. What happens if we denature that protein? We had this 3D configuration, and we either changed the pH or added heat. We broke some bonds, and now we have this protein. Well, you can imagine as molecules are activated by this lock and key enzyme thing, they only fit into the enzyme in a certain way. And so this molecule right here has an unusual configuration. And so this unusual configuration would fit into that enzyme to get it to interact with another molecule. Well, you can see this protein, which has been denatured, has an entirely different molecular arrangement, three-dimensional arrangement. So I obviously wouldn't fit into the enzyme. So this protein is now non-functional. Classification of proteins, shape and function. There are fibers, which are strand-like proteins, structural movement, collagen, keratin, spectrin, elastin. There are proteins, fibrous proteins responsible for movement, actin and myosin. When we get to chapters 9 and 10, we'll talk about those. Muscle contraction. We have globulins, okay? Transport and membrane pumps, regulating pH, regulation of metabolism, body defenses, antibodies, and protein management. Molecular chaperones aid with folding, repair, or breakdown of damaged protein. So these are some of the functions of globulins, okay? And then we have catalyst enzymes that control all reactions by reducing the activation energy. This is what happens with this is an enzyme here. These are two molecules that are going to try to interact with the enzyme. These molecules have a certain configuration and they fit into the enzyme. They fit into the enzyme. Once the enzyme has these two molecules attached, the enzyme will then undergo a configurational change, and it will bring those two molecules at the bonding site close together, and then a bond will form, okay? Then this molecule will be released. These molecules will be released here, bonded. The enzyme, nothing happens to the enzyme. The enzyme then goes back and performs this again. Important in fit of enzyme to substrate. Substrate equals molecules being acted upon by the enzyme. So these are substrate, this is the enzyme, okay? Lock and key model, believe there are different enzymes for every reaction of the body. What does an enzyme, what happens to an enzyme? Or why do we need enzymes? For a reaction to go, okay? For us to have a reaction to go from reactants to products, okay? For a reaction to go reactants to products. So here's an example. A plant takes CO2 and water and makes glucose and oxygen. So the reactants are CO2 and water. The products are glucose and oxygen. It requires energy, okay? Activation energy has to be required. That activation energy is in the form of photons of sunlight. So that's an example of an activation energy, okay? We get that externally, okay? We get that externally. Imagine inside our body, okay, plants also use enzymes, but imagine inside of our body, we've got trillions of reactions going on. We're making ATP at a phenomenal rate, but we would like to use less energy to start these reactions, to go from reactant to product. So rather than using this much energy, you know, from the reactant to up here to get the reaction started, we'd like to use less energy, okay? If we can use a substance that actually brings the molecules that are going to be reacted upon in close proximity, that would lower our activation energy because we wouldn't have to expend that energy getting those molecules together. And so what an enzyme does is lower the activation energy, lower the energy that's required to go start from the reactants to get the reaction started to a point where the reaction is going to continue to form products. And that's what an enzyme does, lowers the energy of activation. That's this energy of activation here. Enzymes can't force a reaction, so if the reaction's not going to go, the enzyme's not going to make it go, okay? Or it can't change the reaction in any way. And this is just a bigger schematic. Here is the substrate. These are, so in this particular case, these are two amino acids that we want to put together, interacted by an enzyme. So they plot down an enzyme in a certain way, because remember, we're going to get the side groups to interact to undergo dehydration synthesis so we can put them together. We then have them interacted. So we have the substrate and the enzyme. A configurational change goes about. Energy is absorbed and a bond is formed, but we require less energy now because now we have them, essentially right next to each other. Water comes out because we do a dehydration synthesis. The product is then released. We have now a dipeptide. This is called a peptide bond. Anywhere we put two amino acids together, it's called a peptide bond. And the enzyme is released. A 3D shape is disrupted. The enzyme is no longer functional. Enzyme's a protein. A protein's an enzyme. Okay, proteins can be enzymes. They are obviously have a very specific shape, and that shape is related to what substrate they're going to interact. If we denature the enzyme in whatever way, we talked about heat and pH the other time, it's no longer functional. Last category of macromolecules we're going to talk about is nucleic acids. They contain, remember, carbohydrates contain carbon, hydrogen, oxygen in 1:2 to 1 ratio. Lipids contain carbon, hydrogen, oxygen, but not in a 1:2 to 1 ratio. Amino acids contain carbon, hydrogen, proteins contain carbon, hydrogen, oxygen, and nitrogen, and sometimes phosphorus and sulfur in the side groups, but always carbon, hydrogen, oxygen, and nitrogen because the nitrogen's in the amino group. Now we've got nucleic acids, carbon, hydrogen, oxygen, nitrogen, and phosphorus, okay? And phosphorus. So nucleic acids contain these particular atoms, okay? Structural unit is the nucleotide. Nucleotide consists of, okay, a ribose sugar, okay, a ribose sugar, and a phosphate group, and one of these nucleotides. Okay, this happens to be DNA because if we look at this 5-carbon sugar, okay, we don't have an oxygen here, so this is deoxyribose. So we have deoxyribose, we have a phosphate group, and we have adenine here. Okay, DNA, deoxyribonucleic acid, consists of deoxyribose, the phosphate group, and one of these nucleotides, adenine, cytosine, guanine, or thymine. Okay, this happens to be adenine, this happens to be thymine. You don't have to remember the structure, you do need to know the names of them and know that adenine binds to thymine and cytosine binds to guanine. And look at this, they bind by hydrogen bonds. They bind by hydrogen bonds. So we have 2 hydrogen bonds here. And notice on this side, we have a 5-carbon sugar, we have a nucleotide, we have a 5-carbon sugar and a phosphate group. So this is the double helix. This is one strand of DNA. This is a strand of DNA with a single strand going this way, a single strand going that way. So this is DNA, it's a double helix. Found deoxyribonucleic acids found in the nucleus, it's double-stranded. It provides directions to build every protein in the body. So every cell in your body, doesn't matter if it's your big toe cell, your liver cell, or your scalp cell, has the directions in the nucleus to build every protein in the body, whether it be an enzyme, whether it be a steroid, whatever it is, it has the directions to build it in every cell in the body. There are signals that make it into the cell that's supposed to be, but we're in the process, and you will in your lifetime start manipulating cells. We're already doing stem cell therapy in the heart and spinal cord and other places like that to regenerate tissue where the tissues don't normally regenerate. It replicates itself. Obviously, if we're going to divide the cells, it's got to replicate itself. And the base pairs are adenine, thymine, cytosine, and guanosine. So everywhere you have an adenine, you're going to have a thymine on the opposite side, adenine, thymine. Everywhere you have a cytosine, you're going to have a guanine on the opposite side. So this is the way they bind. This is what so these are the things you need to remember about DNA, okay? RNA is ribonucleic acid. The big difference, there's three big differences there. Number one, it's single-stranded. Number two, instead of thymine, it has uracil. And number three, it has ribose as its sugar, okay? It's ribose as its sugar, not deoxyribose. So the 5-carbon sugar is ribose. And number four, there's actually a fourth difference. It can be located outside of the nucleus. The DNA is primarily 99.9% inside the nucleus. Mitochondria, as we will learn in cell biology, actually have their own DNA and RNA. So that's one instance that DNA is outside the nucleus. But other than that, DNA is located in the nucleus. RNA can be located in the nucleus or outside the nucleus. They are three primary types of RNA. There's messenger RNA, there's ribosomal RNA, and there's transfer RNA. Messenger RNA is going to carry the message or the blueprint from the DNA as to how to build a protein. Ribosomal RNA is going to be the site of where the protein or the steroid or whatever needs to be built is going to be built. And transfer RNA is going to bring the individual amino acids needed to build that protein. So there's three types of RNA: messenger RNA, ribosomal RNA, transfer RNA. Adenine phosphate, it just so happens that adenine phosphate, which we talk about as a nucleotide in DNA and RNA, if we add two more phosphates for it, we get adenine triphosphate, okay, adenine triphosphate, and that's ATP. That's the source of all the energy in the body, the source of all the energy. When we do cellular respiration, we break down glucose in the presence of oxygen. We get CO2, which is blown off in the lungs, water, that's metabolic water used in reactions, and we get ATP. And when you are in anatomy and physiology 2, we'll discuss that in more depth as how many ATP we get. So we have three phosphates here, okay? The high energy phosphate is the last phosphate. The high energy phosphate is the third phosphate. In ATP, that third phosphate is taken off. Energy is released to drive reactions, drive all the reactions in the cell. And then that ADP, adenine diphosphate, is then rephosphorylated, okay, to add a third phosphate to it to gather more energy. That's the source of the energy in the body, okay? When the body needs energy, it breaks ATP into ADP plus phosphate, and the energy is released and available for use. The body then takes energy from glucose, breakdown rebuilds ADP into ATP. So we store the energy from glucose into ATP, and then we put it back into ADP when we break down more glucose. And so that's how that goes. It's used in a whole bunch of things to transport stuff. If we've got to do active transport, you know, if we have to transport stuff inside and outside the cell, then we use energy. If we do a muscle contraction, we do use energy. Sometimes if we're putting reactants together with products, we require energy, okay? So there's a number of areas that in the body that energy is used, okay? In summary, inorganic compounds are non-living, organic compounds are living. Inorganic compounds usually contain no carbon. Organic compounds contain carbon and hydrogen. Inorganic compounds, as a general rule, are ionic bonds. Organic compounds are covalent bonds. The ionic bonds are, an ion is an element or group of elements that has gained or lost their electron, and because of that has either a positive or negative charge and is attracted to one another. Covalent bond is sharing of electrons. Inorganic is smaller molecules. Organic is larger molecules. Inorganic is water, salts, acids, and bases. Organics is carbohydrates, lipids, proteins, and nucleic acids. So we have inorganic chemistry, we have organic chemistry. If you're a chemistry major, you're going to take inorganic chemistry, which is it's Chem 101, 102. In my day, it's 1000, 2000, whatever it is here. And then you'll take a meshing of these, okay? which is called biochemistry. And biochemistry includes biochemistry, the chemistry of life. So it includes these organic molecules here, includes this organic molecules, and also includes inorganic molecules because the electrolytes, sodium, potassium, chloride, those are molecules of life. Water is certainly a molecule of life. So there's a number of branches of chemistry, and these just happen to be 2 branches of chemistry. And that's the end of this lecture.